A very well welcome to all of you who have joined us today. A very good afternoon to all our participants joining us from India, and a very warm welcome and good morning to all our participants joining us from Europe. My name is Samrat Kumar, working for EURAXESS, and this is our third out of 5 events on the EU-India co-funded call series. And today, topic is very, very interesting, but also very extremely relevant for all of us. It's about the pandemic preparedness and response. And it gives us an immense pleasure to bring to you some very interesting experts on this topic, but also we're very much looking forward to the presentations we have from our research community, both in India and Europe. So without any further delay, I would like to ask Mr. Pierrick Filon-Achida, who is Counselor, Head of Research and Innovation at the EU delegation to India, to give the opening remarks of today's session. Pierrick, thank you so much, and the floor is yours.
Hello, good afternoon to the colleagues in India and good morning to the colleagues in Europe. It's a pleasure opening the session today. I am based at the EU delegation in India, in Delhi. And today we have put these programs with the support of Dr. Samrat and Dr. Vivek and many other colleagues who are speaking in order to facilitate a very difficult step, which is to bring together the European and Indian teams for a proposal. You will discuss today ideas, which is good, but there is a big challenge behind. It's how to make a proposal. So in the program today, we will start a little bit with this flavor of, and Dr. Vivek, my colleague, will present how to put a proposal together, what is required. And after, our hope is that today, with an impressive list of colleagues from India, whom I would like to thank, all the colleagues from Europe who are going to pitch, is to present some ideas. And we hope that some of those ideas will be realized in the form of proposals. It's not the first time that we have EU-India cooperation. So in fact, there are many projects, especially in the domain of health. So you might look at those projects if you know some of the existing coordinators, existing Indian and European participants. You can ask your peers. Just to give you a few messages, EU, my office, and DBT on that specific call are joining hands. It means that DBT will be able to finance the successful and retain Indian proposals with Indian partners in the EU projects. On our side, on the EU side, the European Commission will finance the European. So it's a difficult attempt, but thanks to the support of the ministry, thanks to the support of DBT, we are able now to announce that the call is there and money will be available. A second message is how you put a proposal. So as I say, Dr. Vivek will explain, will help. And furthermore, Dr. Samrat, who is the head of EURAXESS India, will be after this meeting sharing with you information who has been coming today, because it's a first step. You have also a deadline which must be considered for application. You have also to consider that today you will discuss very precise ideas with the European and Indian colleagues, but there will be more calls coming. So we will keep you informed because we have your email and we believe that you are highly interested in all those calls. So when you present a proposal, don't be disappointed. It doesn't necessarily work the first time. You have to do it and redo it and redo it. Last point is that Dr. Manish Maini was supposed to speak. He's from DBT, but unfortunately he's traveling and he cannot connect. So
Manish has connected. Manish has connected. Yeah, finally.
Okay, so this is a very good news, and I will remain to listen to what he has to say. So in conclusion, number one, thanks to all the colleagues today, it's very, as we say in government, down to earth. You have to connect with your pals. You have to put the best proposal as possible, and we will help you in that process. It takes a a substantial amount of time, but once you know how to do it, it increases your chance. And it's also a big family around the table, so it's a way to know each other. We are today speaking only on projects proposals, but the EU, and I want to make it sure that you also get that message, has many other schemes. We have mobility schemes of fellows, of scientists. So it's not the purpose of today, but we can send master students from India to Europe, from Europe to India, PhD students, postdocs, scientists. We have also a huge mobility program. So it can be complementary. It means whatever good ideas you have today, don't throw it away, keep it and develop it. So that's it. All the best. Thanks for everybody behind. I cannot give all the names in Europe and in India, and I will pass the floor to Manish, Vivek, if he's connected. All right. Perfect. So all the best. Have a nice day of exchange. Because of GDPR, we have to find a way around, but during the meeting, we are able to share contacts. Otherwise, we will have to go asking each of you, do you agree, and so on. So we will do this seamlessly today. Thank you very much. Back to you, Samrat and Vivek, for the logistics. Thank you.
Thank you so much, Pierrick, for your opening remarks and really highlighting the importance of this cooperation, but also the possibilities which are out there. Like, it's a huge opportunity for networking also and being part of the EU India Research Innovation family. And of course, it's a tough call, but it's very competitive. But there are big chances, to get funding if one joins hands between Europe and India, specifically for this call. And now I would like to ask Dr. Vivek Dham, who is the advisor at the Research Innovation section at the EU delegation to India. He will now introduce the technical aspects, the guidelines for participation for this call, both from the EU and the Indian DBT side. Thank you so much, Vivek, and floor is yours.
Thank you, Samrat. May I request Manish, you are able to connect? Are you able to give some keywords to the participants? Manish? I think you are mute. I think he has a technical problem. So maybe in the closing, if you can manage to find a solution, then we will take over. So thank you very much, Samrat. I will just quickly go over the modalities of cooperation for this particular call. And just a minute. Samrat, could you please confirm my screen is visible and I'm trying to make it just a minute.
It's visible.
Yeah, I will just try. Okay, hide. Okay. I hope now it works properly. It is visible. Okay. Good
Perfect.
afternoon. Good afternoon, everyone. I'm going to give you an overview of modalities of cooperation. This is a joint presentation between the EU and the DBT. And in next 15-20 minutes, I will explain you how the modalities of both the sides are agreed and planned. So this is the content where I will just quickly go over the different eligibility criteria, admissibility of the proposals, how to submit the proposals, what are the funding rules of Government of India and European Commission, how do you form the consortium, what are the regulatory and ethical considerations you should consider at the time of preparation of the proposal, and evaluation and timeline. So this is what I will cover in next 15-20 minutes. So as Dr. Samrat already mentioned, the call is already open and it will close on 13th of April. So you have very short time to prepare a proposal. So this is what the main challenge to how to find the best partners to formulate a good consortium. And this event is an opportunity for you to meet each other, exchange ideas, exchange the contact details, so that you can try to find a proper partners who can be interested to put forward the proposal. So to begin with, all the information related to this call, it could be the scope, impact, implementation of the call, and the specific eligibility of the European side is should be referred only and only through Horizon Europe funding and tender portal. So this is your first keyword, funding and tender portal, where all the information about the call is available. There is a deadlines. The application submission link is also available on this particular website. So please go through that. At various web pages and the sites, the link to specific call will direct you to the funder and tender portal. So DBT, I think their web notice will be published today on their website. So please check it by 5 o'clock or tomorrow morning. So it should be live. So which gives an exclusive information about what are the DBT's rules and regulations and ethical clearance and the funding annexures they want. So this is a very comprehensive guidelines they had prepared, and we are very happy that each and every aspects of putting forward the proposal is addressed in this particular website, the web notice. So please go through the web notice very carefully. You will find all the information what you are required to. If not, then we are more than happy to help you out. So who can participate? Basically, this program, the Horizon Europe program, is by default open to almost all countries. Any legal entities, can be a big company, small companies, startups, SMEs, research organization, universities, non-governmental organization, who has a capacity to address, to solve the challenge which has been put forward in the call, is most welcome to be partner into the Horizon Europe. Okay, so that is the requirement, or that is the participant conditions from the European side. From Indian side, the government of India will encourage participation from center and state government supported organization. It could be public or private academic institutions, universities, research institutions funded by national or state institutions, or R&D labs, or state universities, deemed universities. All these people are eligible for participation. The government of India and DBT also encourage the participation of private entities. It could be SMEs, small and medium sized sectors, as well as the startups, but on their own funding. I will come to that point later on. So this is the basic eligibility condition. I may repeat this particular condition again and again, because this is the basic admissibility condition of Horizon Europe. So you need to have at least 3 European partners from 3 different countries. Okay, at least one partner should be from European member state, and 2 could be from the member state or associated countries. So here is the list of 27 European member states are given, and along with this 27 member state, Horizon Europe, there are 16 countries are already associated with it. Okay, so you have a bunch of almost 44 46 countries to choose a partner. So normally this is the multilateral program, so you need to have more than 3 partners into basically minimum 3 partners, and it could be 5, 10, 8, 15. On an average, Horizon Europe consortiums are having 8 to 15 partners. So that is the normal size of the consortium. The participation of India is not a prerequisite for this particular call, but to be, that is the Horizon requirement. Okay, but if you want to be the DBT's co-funding beneficiary, you need to have one or more Indian entities to be part of this particular proposal. The DBT also encourage more than one entity, because that increase the impact of particular project in Indian context. So out of, if there are more than one entity from India, one should be from publicly funded entities. That is what their uh, sorry, that is what the basic requirement of the call is. And apart from that, you need to have 3 Europeans partner, one Indian partner or more Indian partner, and plus you can add any entities in non-European countries. I will come to that point later on. Please check additional eligibility condition in the call text as well. So this is what the call text is all about, and the the eligibility condition specific to call is also given. But there is a very important point which has been mentioned, and my colleague from Brussels will also elaborate on this particular point. The WHO list of priority disease identified should be considered at the time of your proposal formation, and there is also ERA list of identified uh, diseases has been given in the list of this particular call. So before you enter, please go through the list which is given in the call text. So as I mentioned, who is eligible for Horizon Europe funding? Basically, 27 member state plus 16 associated countries, and plus there are 120 lower or middle income countries are eligible for the Horizon Europe funding. So, but there are almost 10 to 15 countries which are considered as higher economies, developed or emerging economies. They are not directly funded or don't receive a funding from European Commission or from the Horizon Europe program. So for that, we have created a co-funding mechanism. Basically, non-European countries are listed here, that is US, Canada, Australia, New Zealand, Japan, Korea, China, India. You name these big countries. All these big countries are not funded, but they are bringing their own money from either own resources or from the government. In today's call, we only focus on co-funding possibility, because there are three different scenarios under which the co-funding can be provided. One is exceptional funding by the European Commission, co-funding, and specifically mention if the country is mentioned in the call text. But today's call is co-funded by Government of India, so we only finalize or fix our attention on the co-funding mechanism. So the Indian entity who is going to participate in this particular call should compile the terms and conditions of DBT, which is very well explained. The proposal should be jointly formulated and submitted at both the agencies. That is, same proposal which you will submit at the European Commission, the same proposal should be given to the DBT as well. The positively evaluated proposal by both the sides will be co-funded by DBT. The Indian side will not sign European grant agreement. Instead, they will sign an associated agreement. I will come to that point later on. And the duration of the project proposal could be 3 to 4 years. So who is eligible for DBT funding? As I already mentioned, academic institutions, it could be university from central university, state university, or private universities, research institutions, national laboratories, state-run laboratories, which are publicly or privately funded, are eligible for the Government of India's the DBT's funding. So these institutions normally has PFMS, that is the very standard registration system which should be in place. And in addition, if the industry want to be participate, they are highly encouraged, but they have to bring their own funding. Okay, so the private entities could be SME, could be big industry, or startups are not directly funded by DBT. And this particular co-funding is only available for the 5 calls which of the Horizon Europe, which has been earmarked by the DBT. The funding from the DBT is basically a grant in aid. You can, this is very standard cost of eligibility costs, manpower, consumables, travels, overheads, all these are covered under this particular grant. The DBT will contribute or fund the entity as per the requirement of Indian entities project participation. So there is no cap. They will assess how much money is required and how much money has been asked by the Indian entity. As I mentioned, the proposal has to be jointly developed by European, Indian, and other partners. They're in the Horizon Europe template. So the template had part A and B, and this particular template first submission will be at the Horizon Europe funding and tender portal. The same proposal you will submit to DBT along with the financial and administrative annexes which are given on their website. You will submit, that is the additional detailing of the budget is required. In addition, once you submit the project, the European coordinator will submit the proposal to Horizon Europe funding and tender portal. You can submit that proposal along with the additional documents, that is funding, the budget document, to DBT within 7 working days. Okay, so you have a grace period of 7 working days to submit at the DBT. The proposal has to be submitted via email. So make sure your proposal part A, B, and the annexures are visible, are printable, and can be opened easily. So this is what the submission process. First part of the submission, part A and B, is a very standard done electronic procedure by the European coordinator. And the second part, that is DBT submission, the part A, B, and plus the annexes of the India's budget format should be submitted to DBT through via email. The email address has been given on their website as well. So the first very important point is how to form a consortium. Basically, as we are the funders, that is the DBT and European Commission, we are only providing the platforms for the researchers to identify potential partners. So these are the networking brokerage events we are regularly organizing. So today's brokerage event is focused on the specific call. So actually we are organizing brokerage event for each call which we are going to co-fund. In addition, the European Commission also organizes regularly info days at European Commission through virtual platforms. NCPs in Europe are also doing the networking job and assisting it. EURAXESS is also providing a support to identify the partners. So consortium formation, again, 3 legal entities from 3 different European countries is the prerequisite. In additional entities can be highly encouraged. That means the Euro-Indian can be 1 or more Indian can participate into this particular proposal. The project proposal should be, multidisciplinary, cross-sectoral, cross-border is highly encouraged. Gender balance should be maintained. One project coordinator should be from Europe, because the entire coordination of the project should be handled by the European coordinator. And if there are more than one legal entity from India, so you should nominate one legal scientific coordinator which can communicate with the DBT. So the Indian regulatory and ethical considerations, this is very important part, because we had in past experience that people are not very aware of this regulatory and ethical considerations, and that is why the projects got delayed. So make sure when you are identifying a lead Indian coordinator, he or she should be well aware of the DBT's the government of India's ethical clearance process, and it should be start as soon as possible when the project is given an what you call the success award has been communicated to you. So DBT as well as the European Commission also encourage people in the consortium, the PIs, to pay attention to IPR. This is most important because these are all projects having an commercial values, and output from this particular projects are shared by different entities. So make sure IPR are well sorted out at the consortium itself. Application form, as I mentioned, application form has 2 parts, part A and B. That is the template of Horizon Europe you will apply. So the Indian entities will participate as an associated partner. That means they will receive the funding only from Government of India. They will not sign association grant agreement, the grant agreement with the European Commission, whereas the European entities will be considered as beneficiaries and they will be signing the European grant agreements. So this is what you will communicate on day 1 to the European coordinator or the consortium members, that the Indian entity should be considered as an associated partner, and you will only be signing a consortium agreement. This is what the message you should give on day 1, and accordingly the proposal and your participation should be considered in the project proposal. the work, you cannot coordinate the project, but the Indian entities can lead work package and could be partner. To be part of this project proposal, you need to have the Horizon Europe participation identification code, which is very standard and which is most of the Indian institution has. If not, the link will be given at the end of my presentation. So the Indian entities will, as I mentioned, will be participating as an associated partner. So you don't have to give any budget details. Okay, you will only give a lump sum amount, how much the amount in euros is required or is planned in your proposal, and that amount will be mentioned in part A of the Horizon Europe in euros, and that will be done by the European coordinator. So the evaluation criteria, there will be two-step evaluation. The first evaluation will be done by the Horizon Europe as per the peer review process, that is excellence, impact, and implementation. This is done entirely by European Commission, and European Commission will inform Government of India the successful entities or the proposals with Indian entities in it, that these are the proposals has been successfully shortlisted for the grant or the main listed, and those proposals will be re-evaluated by Government of India as per their national regulations. So the proposals which is evaluated by both the positively evaluated by European Commission and the Government of India, in that proposal, Indian entities are eligible to receive the DBT's funding. Okay, so if the DBT don't evaluate positively, the European Commission will continue with the proposal as per the Horizon rules and regulations. So as per the timeline, the proposals already, the calls are open for the 3 months, then it will be closing on 13th of April. From the closing of the call till the grant agreement, we are obliged to complete the entire process within 8 months. The evaluation will be done within 5 months, and then grant agreement for next three months. We anticipate the Government of India will do the same, follow the same line, and the project will be start within the 8 months after the closure of the call. So these are the different websites where you'll find different platforms to identify the partners, how to find the old projects proposals which can help you to identify different European and Indian entities working in the particular areas. So as I mentioned, this is entirely, the European system is entirely electronic system based. So you need to have a EU login. So this is the steps which is also given in the DBT's website. You will also have a participation identification code, which is very standard, and it is nicely explained in European Horizon Europe tender and funding and tender portal. So this is, if you need any help, we will be more than happy to help you out. Apart from that, if you want to put a proposal or not, okay, whether you want to be part of this consortium or not, if you are not going to put the proposal, we are looking forward to receive or to integrate a lot of international experts to evaluate these proposals. On an average, we receive 10 to 15,000 proposals per the calls which we are receiving. We are publishing something 700 calls in this particular work program. So we need a lot of expertise across the globe. If you think you have a skills, you have expertise, you can register in our database and you can be called as an expert to evaluate the proposals. This is not a free of cost job. The European Commission funds quite handsomely to the evaluators. On an average, 3 to 4,000 euros per evaluation is paid by the European Commission. So this is another way to get to know how the multilateral collaborations are working. And we will be more than happy if you need any assistance about the clarification of the participation. My team from the European side or the DBT sides will be available. These are our email address, and I will stop it here. If you have any questions, we will be more than happy to answer. Thank you very much.
Thank you very much, Dr. Vivek. Again, a very clear, structured overview about the technical aspects of how to participate in this co-funded call. I don't see any, I see, yeah, there are some questions in the Q&A, Vivek. Should we just take them? There's one question, I'm not exactly sure about that. What are the priority pathogens for the call?
Yeah, so I think Dr. Stefani will give you more detail, but in my presentation and the call text, there is already a link of priority pathogen, which has been given in the list of WHO. Okay, so the link is already given, but the next presentation will elaborate specifically about this particular aspect.
Okay, perfect. Because also the second question was from the same participant asking about the pathogens and the list of on the WHO website. So we will just
I will share that in the chat as well. So the website, I will share that. Yeah.
Okay, perfect. I think with that we could go to the next part of our event, which is presenting you the scope of the call, and we are very happy to welcome Dr. Stefanie Sowinski. She's a policy officer at the Directorate on Health and Research at the European Commission, joining us from Brussels today. And thank you, Stefanie. Welcome back virtually to India. We had you a couple of weeks ago when we kicked off the Horizon Europe Info Day. And so we're very happy to have you back again. And today you have a very interested audience, curious to know more about the scope of the call. Thank you so much. The floor is yours.
Perfect, thank you. I had a pleasant journey, I can say, to India. I'm supposed to share my screen, is that correct?
Yes, please.
Okay, I will do so, and you let me know what you can see. I'm gonna try to move this bit. Is this wait a moment.
Perfect. We can see the full screen.
Can see it?
Yes.
Okay, great. Okay, so the call I'm presenting to you today, that's from the cluster 1 that's the health cluster in Horizon Europe, and the top. Oops, I don't know why it's advancing on its own. Sorry, and the topic basically aims to advance the development of broad-spectrum antiviral therapeutics for infectious diseases with epidemic potential. So we obviously all know that the emergence of viral diseases will increase in the future. And because of that, the availability of broad-spectrum antiviral therapeutics will provide a critical preparedness and response measure against these infectious disease epidemics or pandemics even. So the total budget of the call is 50 million euros, and we estimate that the expected outcomes can be reached with an EU contribution of 7 to 8 million euros per project. And proposals that are submitted under these topics are supposed to deliver results that can contribute to the expected outcomes that are listed here. So these are that the scientific and clinical community has an increased knowledge on the viruses with epidemic potential, and in particular, a better understanding of different mechanisms of action for the development of broad-spectrum antivirals. The community has access to novel approaches for the development of antiviral therapies for emerging and re-emerging infections, and the scientific community has access to experimental broad-spectrum antiviral candidates that can be used for further clinical investigation. And we have established a diverse and robust pipeline of broad-spectrum antiviral drug candidates. So then the scope of this topic, so proposals that are submitted to this topic, they should develop and advance broad-spectrum antiviral compound and as well as novel approaches to the development of such compounds. And they should target viruses with high epidemic and pandemic potential for the EU, and those are the ones that are listed in the WHO list. And then there's also a list that was basically authored by the Health Emergency Preparedness and Response Authority, HERA. And they have listed some criteria that the link to these criteria is on the slide and also on the core topic, but I'm also happy to tell you later more about it. And the proposal should cover viruses for which there are no currently available effective therapeutics, or for which the therapeutics are suboptimal. And they expected to incorporate state-of-the-art screening technologies and innovative approaches to identify new targets for these antiviral compounds. And the emphasis here is really on broad-spectrum antivirals, but it might also include repurposing of previously approved or in-pipeline drugs. So important to say here is that the proposal should really address all of the following areas. So all of the bullets that are listed here. We want proposals that include preclinical work, proof of concept first in human studies, and early safety and efficacy trials for these compounds. Phase 2B and phase 3 trials will not be supported. We want also proposals that look at innovative delivery systems and suitable safety profiles for broad use when this is possible or relevant, applications of novel approaches and widely applicable workflows, for example, artificial intelligence, for the rapid identification of those antiviral compounds should be included, and attention should also be paid to the social factors in the relevant part of your scientific actions, like sex, gender, age, and socioeconomic factors, ethnicity, migration, and disability. And one important note is as well that last year in November, we had a workshop that was organized by the Commission, and that was looking specifically at the development and availability of broad-spectrum antivirals as a priority medical countermeasure. And we have a report that summarizes the outcomes of this workshop, and the link is in the slide, but it's also in the frequently asked questions when you go to the funding and tender portal for this core topic. Then for your interest, I have listed 2 additional topics here on the slide that are also within the scope of pandemic preparedness and response. And this is the understanding vaccine-induced immunity topic, and also the topic on immunogenicity of viral proteins of viruses with epidemic and pandemic potential. But important here is that these topics are not co-funded by the Indian government, but Indian entities obviously are welcome to participate, but there will be no co-funding. And that's it for my side then. Happy to hear your questions. Did I stop sharing?
Yeah, thank you so much. Thank you so much, Stephanie, for your presentation. And let's see if we have any question. Yeah, we have one question in the chat, which is from Dr. Yuvraj Gujar. He's asking, development of broad spectrum, where should clinical trials center, and drug development by use of surface submerged fermentation if needed to use of fungal bacterial origin? Question to Dr. Stephanie.
So that's the first question, I understand. The second one, I need to maybe ask for clarification. So the clinical trial, so what we said in the areas that proposal should address, that we would like to see a preclinical and also early in human trials and efficacy data if possible. So that these are the trials we would like to see. So it's the early phase 1 to A if this is possible. The phase 2B and 3 are not included, but it is up to the applicants to see how best to describe the actions, but definitely preclinical and early safety and efficacy data should be considered in that core topic in the proposal.
Follow-up question.
Stephanie, can you just elaborate a little bit about the list of WHO list which is in the call, but do you want to elaborate a little bit on that?
I have to admit, so it's definitely the list that WHO has, and the link is there, has published. I don't know all the pathogens by heart. You can just click on the link and you will see the viruses in there, what will be considered. The other thing that important here is that HERA has published fact sheets for which the link is also in the call topic. And the criteria that should be considered here are that the pathogen with high pandemic potential, that have a rapid transmission mode, likelihood to reach a sensitive population, for example, persons with minimal pre-existing immunity, and that have a high potential to cause high morbidity and mortality. So this fact sheet also states that these are largely respiratory RNA viruses, but it's important that the scope is not limited to those. And also why the WHO list is put in there, it says in the call topic such as, so again, this is a guidance, but it's also not limited, but obviously the criteria by HERA should be considered. So it needs to be really viruses with high epidemic potential. But the list, please check the link, and then that will be very.
I had already shared the link in the chat with the audience.
Perfect, thank you.
There is one more question related to whether this call looking for the vaccine or a specific active molecules.
So this is on therapeutics. So this is antiviral therapeutics. So treatment, not vaccine. But the other topic I put in, let me just see, that is looking at the vaccine-induced immunity and immunogenicity of viral proteins. So these are two other topics which might be of interest, but again, looking at vaccines, but again, then not co-funded by India. But our topics is specifically on treatments and antiviral therapies.
Yeah, that is very important point, because we keep on receiving a lot of emails related to that, and thanks for that clarification. Thank you.
Yes. But the type of compound, sorry.
No, please go ahead.
I just wanted to say, because I received the question already, I think also from you, the type of compound is not really specified. So it can be small molecules, but it could also be other inhibitors you can think of, or antibodies, yeah.
There's one question, Stephanie, on publications. Should we have prior publication in these proposed areas, or can we initiate studies using the funding?
So for this, it's preclinical work, discovery work is included, but then the independent expert will obviously also evaluate the excellence part of the proposed work. And sometimes that then will be good that there has been already some experience by the group in this area of work, but that will be really up to the independent experts who will evaluate to see how sound the concept is of the proposed work under excellence.
Okay.
That is a good question now about the deadline. I don't actually know the deadline, sorry, but it's on the website as well.
Yeah. Maybe Vivek, do you have the deadline in mind?
You're muted, sorry.
Sorry, the deadline is 13th of April. So it is mentioned almost every part of the web notice of the DBT, the call this, as well as the Horizon Europe website as well. The second point, there is again a question in a chat about the Zika virus. I think that is, can you elaborate, Stephanie? Is that, I think it is in the list I saw quickly.
Yeah, I think so too. Yes,
Yeah.
it's in the but yeah, please check the WHO list, and yeah. If it's also rapid transmission mode, likelihood to reach sensitive population, if these criteria are in, then yeah.
That's true.
Okay.
And Stephanie, just whenever there is an info day about health, please keep us informed so that we can also bring on the Indian audience into the info day in Brussels. So that is a virtual event. So that is also a good occasion to meet different people from different country.
Okay. You have the recording of the last info day, I'm assuming.
Yeah.
Okay.
We
Perfect.
do. Yeah.
Perfect.
Okay.
Also, any questions that follow in written, I'm happy to answer these in written as well. And can look more into the other questions.
Yeah, thank you for that, Stephanie. And as Stephanie said, if there are any questions which hit you maybe after the webinar, you can always address them to us, and we can forward them to Stephanie or to the appropriate contact in the delegation or in the commission. I think if we have no more further questions, I would like to thank Dr. Stephanie Sowinski for being here with us, and a happy virtual journey back to Brussels. Thank you.
Thank you.
And we are looking forward, hoping to that there will be some very exciting applications coming across in the coming weeks. And we are now going to the next part of the event, which is now about the pitching the proposal ideas of our scientist community from India and Europe. And these are our flash presentations. With that, I would like to welcome all the presenters who have been selected to present today their project idea and their expertise. Before we start, I just have a very one main concern, which is the time. So please bear the time limit of 7 minutes per presentation. I will be timing it. I've been told by Dr. Vivek to be very strict on that. I'm a bit more lenient, but I have to follow his, in that sense, my superior here in this webinar. So I will have to keep the time, 7 minutes each. And we start with Dr. Murugan Veerapandiyan. He's a researcher at the CSIR Central Electrochemical Research Institute here in India. A very good afternoon to you. Point of need analysis for antiviral therapeutics is his presentation, and he already has opened his screen and sharing it with us. Thank you so much. Dr. Murugan, the floor is yours.
Thank you, Dr. Samrat, for your brief introduction. And thanks once again, European delegates as well as Indian delegates for giving me this opportunity. A warm good morning and good afternoon to one and all presented virtually. So my topic for today's presentation is on point of need analysis for antiviral therapeutics. By the way, I'm a scientist and assistant professor in electronics and electrocatalysis division of Central Electrochemical Research Institute, Karaikudi, Tamil Nadu. So our main area of research focus in SICRI, that is Central Electrochemical Research Institute, is electrochemical and electrical biosensors for healthcare. We focus on communicable disease markers, non-communicable disease markers, also developing phytochemical-based biopolymer as redox active element for biosensor research. Apart from all these diagnostics, we also work on high-throughput analytical systems composed of electrical resistant-based measurement of real-time cell cytology, cytotoxicity, and so on, especially monitoring the cell-drug interactions
I think we lost Dr. Murugan. Dr. Murugan, I think we lost you for some seconds. Could you again share your screen? Yeah. I think we lost you for 30 seconds or so.
Okay. So now my screen is again visible?
No, not yet.
Sorry. I don't know what happened. Yeah, just a minute. I hope now it's fine.
Yep. Yes. Now, yeah, you can continue.
Yeah. Sorry about the technical glitch. So our focus areas are related to communicable disease markers, non-communicable disease markers, and electrochemical biosensor materials derived from phytochemicals in conjunction with the biopolymers like metal-free sensor element for various biosensor applications. We largely work on various bioreceptors like antibodies, enzymes, biosimilars, and nucleic acids for various applications. We do have a sophisticated system capable of sensing the real-time cell motion, adhesion, cytotoxicity using electrical resistance-based impedance sensing systems. So our electrochemical and electrocatalysis divisions predominantly working on development of various electrode materials which necessarily exhibiting the redox behavior for biosensor constructions. And one prime interest is without using external mediator, we would like to develop sensors so that the user can, in a simplified manner, demonstrate their portable applications. And we also use customized bioreceptors and bioaffinity layer for amplified signal transductions. In terms of label-free diagnosis, user-friendly are the prime requirement. Of course, detecting the analytes at lowest detection limit without any cross-reactivity, in other terms, anti-interference, and exhibiting improved sensitivity and exhibiting wide linear range of detections are the prime challenges or opportunities involving in biosensor where we are contributing a lot. So we also have portable devices already demonstrated in various know-hows or scientific publications constructed of voltammetric technique, impedometric technique, and amperometric techniques. As you can see, we develop electrode materials, immobilize various bioreceptors, and check their electrochemical behavior with respect to target analyte interactions at the bioreceptors embedded on the electrode surface. So we do have recent contributions in COVID diagnostics, especially customizing the oligopeptides that is relevant to human angiotensin converting enzymes 2 and targeting the receptor binding domains of SARS coronavirus spike protein in association with ICMR NIRT and Dr. MGR Institute for excelling the RNA isolation and amplification free detection of COVID causing SARS coronavirus spike proteins. So the results are already published in Sensors and Actuators, which is a prestigious journals in the biosensor field. As you can see here, we have customized carbon screen printed electrodes with the cost efficient redox active electrode material and customized the oligopeptide specific to SARS coronavirus spike proteins, and we have tested the receptor binding affinity at their electrode surface for lowest detection limit, as well as exhibiting the anti-interferent property. So in terms of antiviral herbaceutical related activity, we do have sensing platform capable of sensing the specific antiviral component in Nilavembu Kudineer, which is known as dengue, anti-dengue treatment formulations in herbal medicines of South India, especially Siddha medicine. So we have commercially procured Andrographis paniculata herbal thing, and we have made alcoholic extract, and we have analyzed through conventional HPLC and LCMS analysis to identify the antiviral component, which is known for exhibiting the specific anti-dengue property. And we have developed a sensor platform which are capable of specifically analyzing the andrographolide. So this is one proof of concept for demonstrating electrochemical approach as an alternative analytical technique for screening antiviral stuff for sensing samples, maybe of pure drug or QC/QA applications of formulation in terms of polyherbal, or measuring the excess usage of antiviral components in various biological or environmental samples. So the pitching point of this particular presentation or research focus under the pandemic preparedness or customized sensor platform design for rapid active pharmaceutical ingredient testing. That is one sort of expertise we can collaborate, and therapeutic drug monitoring, especially monitoring the primary metabolite, secondary metabolite, or unreacted or unpharmacologically active components in the biological fluids to check the therapeutic drug monitoring ability, and analyzing the ex vivo cell protein interactions with the drugs using electrical cell substrate impedance sensing system, and determining the association or dissociation constant, especially with the drug-drug or drug-protein interaction, and testing, of course, antiviral residues or viral particles from hospital effluents. So these are the potential area of research which can be taken in account for pandemic preparedness and develop various collaborative outcomes for the betterment of global healthcare community. So with this, I conclude my presentation. If there is any questions, I'll be very happy to answer. Thank you.
Thank you so much, Dr. Murugan. And we move to our next presenter, Dr. Luc Hilgers. He's from the Netherlands, representing Lightwax BV. Very good morning to you, Dr. Luc. And
good morning.
Floor is yours. Thank you.
I cannot share my screen.
Okay, so if Dr. Murugan, could you stop sharing your screen?
I have already stopped the sharing.
Okay, now it's stopped.
Let's start again. There it goes. Good morning, everyone. Good afternoon. You can see my screen?
Yes, perfect.
Okay. I think there has been some miscommunication because this presentation will be related to vaccines. If we talk about pandemic preparedness, we think immediately of vaccines, and our role in the development of vaccines is developing new adjuvants which open new opportunities for effective single-dose vaccines. So this is not about antivirals, it's about vaccine. I represent the company Lightwax. I'm one of the founders of Lightwax, together with Peter Paul. And so we are focusing on adjuvants for low single-dose vaccines. The company was founded some 10 years ago. It's a small and medium enterprise, and our mission is to develop better vaccines by better adjuvants. And we are a small company, as indicated. We do a lot of outsourcing and establish collaboration because we have only one component of the vaccine. The other part of the vaccine is, of course, the antigen, the virus, or the bacteria. In our 10 years, we have been able to find financing from the founders, many different grant applications. I will talk about one application which is ongoing, which is also a good testimony for collaboration between European and Indian partners. And we also had a loan of regional fund. So an important project for us was the Indigo project, or it's still the Indigo project. It's a collaboration between European partners and Indian partners. It started some 3 years ago, and it will continue 2 more years. It's focusing on a better flu vaccine, as indicated here. And here you can see that there are several parties involved in India in this project. I will show you a little bit more about that. We are interested in the new molecules that have been used in this adjuvant, not only for prophylactic vaccines, but also for therapeutic vaccines. And we would like to understand what is the mode of action. This gives you an overview of the progress we have made the last 3 years within the Indigo project. So we are talking about the vaccine. We have used or developed the adjuvant technology. Yes, that is the adjuvant, and to start clinical trials, we had to produce the adjuvant in bulk or in small vials under GMP conditions. Last year, we started the phase 1 trial with the flu vaccine containing the adjuvant, and we are now in the phase of preparing the phase 2A. So that is really the status of the adjuvant. We are progressing now into clinical phase. At the same time, we are preparing the tech transfer of phase 1, phase 2A in India, because the others were done in Belgium, here in Europe. And all the tech transfer is ongoing, and we hope that we can do a phase 1 trial this year in India and 2A in the next year. And here you can see some of the partners involved. A few are located in Europe, which is the University of Ghent, and Harmony, which is our CRO, and Lightfax. And the other partners are Geneva in India, CMCV, THSTI, and CDSA. So this product, the adjuvant, is now really in the clinical phase for a flu vaccine. A little bit more about the adjuvant. It's an adjuvant we have developed the last 10 years. Compared to classical conventional adjuvants, it has unprecedented activity. It opens opportunities for a single shot vaccine, which is of tremendous importance in case of a pandemic, that with a single shot you can protect the people. It's a fully synthetic compound. It's stable, it's sterile, and it's also important. It's very inexpensive. We have the quality system in place, so we are able to produce the GMP material. And related to pandemic preparedness, so we can support the development of vaccines by our adjuvant, because it's a versatile technology. It can be applied in different targets with different viruses and bacteria. It's aiming at a low single dose vaccine, so you only need a small amount of the antigen, and with the adjuvant you can have a strong immunity after already one single dose. And we are thinking of, to be prepared for a pandemic, we are thinking of stockpiling of the adjuvant, because it's ready to use material. It's very stable and can be stored, as far as we know now, for many years. So at that time, you are ready to develop the pandemic vaccine. So test material is available. So this presentation is a way to reach out to Indian partners or European partners interested in vaccine development against a pandemic agent. So we have test material available for non-clinical studies, but also for clinical trials. We are able to support different pandemic vaccines, because the technology is versatile, so it will work with flu, but also with other viruses and other bacteria and recombinant proteins. We have a significant expertise and know-how. We have a quality system in place, and we have a tremendous network to develop these products. So we are aiming at stockpiling of the adjuvant, and that can serve many different vaccine developments. The manufacturing is ready-to-use multi-dose vials, and to secure for the future, that's important that these materials are available and can or deliver affordable, available, and accessible vaccines. And here's my email address, so please drop me an email if you are interested in developing vaccines and you have a recombinant DNA product and you would like to have more support or a better vaccine, then we might help you. Thank you so much.
Thank you so much, Dr. Luke Hilgers. And of course,
sharing.
Yeah, thank you so much. Very good to know that you already have you're part of our existing EU-India consortium, yeah. And you're reaching out for this call as well to potential Indian partners. So please get in contact with Dr. Luke Hilgers if you are thinking that this would be an excellent match for you. We move on to our next presenter, which is Dr. Sonu Gandhi. She is scientist E at the DBT National Institute of Animal Biotechnology. Niap, and her title is Biosensing Probes, Theranostic Applications, and Best from Waste. Very good afternoon. The floor is yours. Thank you.
Thank you, Dr. Samrat. I think you can hear me.
Yes, and we can see your presentation as well. Yeah.
Thank you. Yeah. So first of all, I would like to thanks for giving me an opportunity to present the research that we do in our lab. Yeah. So our lab basically focuses on 3 different areas. I'll quickly describe that. First is the biosensing probes. So we have the expertise in developing different nanomaterials that can be used for enhanced diagnostic purposes. And we develop different kinds of electrochemical platforms by developing the antibodies. By developing the antibodies and using the aptamers and peptides. So for this, we have several research work which is going on based on the SARS-CoV-2 based diagnosis or a diagnosis of HIV, CTN1, which is the cardiac biomarker, or the rheumatoid arthritis and aflatoxin detections, pesticide detection, and the JEV detection, which is Japanese encephalitis virus. I will not go into the detail, just briefly describe how we do that. So we fabricate different electrodes, various electrodes by using the nanomaterials, using the nanorods, and then couple the antibodies on its surface and then detect the in-house generated RBD antigens. And so these all work, which we do all in the lab. And so we have a various other fabricated sensors. So this is another one, which is the lateral flow dipstick device. So this is like a pregnancy test kit where you can immobilize the antibodies as the test and control lines and test these samples on site. And you can say at home even that the sample is positive or negative for the presence of the virus. So this kit we have developed in the lab with the sensitivity of above 90%, which is approximately 94% sensitive. And then this work we have done by fabricating the field effect transistor based devices using graphene as the electroactive material and use the antibodies to coupled onto the surface of graphene and recorded the change in resistance over the period of time in the real time manner. Another work what we have done is the JEV. JEV is more often misdiagnosed with the dengue virus. So because the symptoms are same and it is caused by the mosquito-borne illness. So for this, we have generated the recombinant protein JEV-NS1 because that acts as the early diagnostic marker, and raised the antibodies in-house and used it to fabricate on the sensor surface by using the reduced graphene oxide as the nanomaterial and sensed it on the surface of the electrode by electrochemical immunosensor. Here we have tested it on the pig serum samples and have obtained the positive response from the sensor surface without any matrix effect. So after that, we have also fabricated the lateral flow device, which is LFA here, for the JEV-NS1 detection. And we found that it was tested on around 66 positive pig serum samples where the sensitivity was obtained in 97%, and this kit is under, we have filed the patent for the same, and the device is also transferred for the technology purpose in BCIL in DBT. The SARS-CoV-2 device is also, we have tested on 100 patient samples in nasopharyngeal swabs and has given us 94% sensitivity, which is also will be gone soon for technology transfer. This is the microfluidic device which we have fabricated in the lab by using a simple Wattman filter paper and created 3 zones, sample zone, detection zone, and control zone, and used the nanoparticles which were coated with the aptamers. So with this, because the aflatoxin or mycotoxins are present in the milk, so by just spiking on the sample zone, you can say the toxins are present or not by just looking at the color of the paper. So you can see there is a change in the color from pink to purple if the toxins are present. So another area is the theranostic application. So here what we do basically in this work, we have targeted the cancer receptor, which is the UPAR, which is urokinase plasminogen activator receptor. So for targeting that receptor, we have used the gold nanoparticles as the material, and then coated it with the polymer, which is the chitosan, to make it more stable, and conjugated this nanomaterial further with the peptide. This peptide is specific for the UPAR receptor, and also it is labeled with the FITC, so that we can do the imaging and trace the receptor in the UPAR. So this study we have done in vitro system. After that, another cancer-based research we have done in proteases, because in the metastatic stage, lots of proteases excrete into the system. So we have synthesized iron oxide particles and prepared or designed the specific peptide, so which is labeled with biotin at both ends, and the protease cleavage site we put into the middle of this peptide, and the nanoparticles were labeled with the biotin. So there is the streptavidin biotin interaction happens. Because of this, there is the aggregation, and the MPS magnetic signal goes down. But once you put the cells which express the protease, it cleaves this peptide from the middle, and you see that the solution becomes dispersed, that you can see with the naked eye, and also you can see in the system, in the instrument, that the MPS signal goes high. So another study of receptor targeting we have done in the glioma cells. So here we have used C6 glioma cells, in which it is overexpressed in the brain cancer cell. So we synthesized the iron oxide nanoparticles and labeled it with the lactoferrin, because it is known that lactoferrin transferrin receptors are present on the surface of the brain, and then the lactoferrin can cross the blood-brain barrier. With this in mind, we have done some in vitro studies in using the C6 glioma cells, and observed that at the 12 and 24 hours, there was the uptake after labeling the particles with the lactoferrin. Another area in our lab, which we have in Ignite now, is the best from waste. So the things which we throw in the dustbin after use, such as the plasmid isolation, these columns which we throw after one-time use, we just cleaned it and filled it with the graphene nanoplatelets powder and called it as the partition cartridge column. And then we used it for the SELEX procedure. SELEX is the tedious procedure for finding out the suitable or robust aptamers. So by using this column, we have reduced the SELEX cycles of the aptamer selection from 35 to 42, the 10 to 15 cycles. So these are all the characterization data. So this is being regularly used in our lab and is a very efficient column. So these all are the expertise in our lab, and I'm very, very excited to have the collaboration with Indian and EU platform to collaborate on the epidemic preparedness call. Thank you so much.
Thank you so much, Dr. Sonu Gandhi, for your presentation and reaching out to potential partners. We go to our next presenter, Sarah Skublikowska from Tech to Market in Poland.
Yes, yes. Yeah, I just enter my video. Okay. Yeah.
Here you are.
Good morning, good afternoon to all. I'm Sarah Skublikowska from Tech to Market company, and today I will tell you a few words about how we can help you as we are offering our partnership. So let me share my screen. Yes. Yeah, can you see my screen?
Yes, we do. You just put it in full screen mode.
Yes, yes, okay, okay, 1 minute.
Yeah. on the here we go.
Guys. So good morning once again. Today I will tell you a few words about our company, Tech2Market Poland, and how we can help you. So we are Polish SME, which core activities is related to the consultancy and helping strategy, business plans, market analysis, technology transfer, and commercialization. Our mission is connecting technology with market, from idea to application, from concept to innovation, and from technology to market. I saw today a lot of great research today, and our mission is to help take this research into the market. So I think that that for sure we will find a way to collaborate. How we can support you in EU projects? Our core activities within EU projects is partnering in core activities. So exploitation, dissemination, communication, IT elements, and also IPR management. So let's talk a little bit more about this core activities of us. So when it comes to the exploitation, it means for us, first of all, in case of high TRL projects, two-stage market analysis with, first of all, basic desk research about key players or overview of situation, market data, expert interviews, and then in the second stage, based on the results of first stage of analysis, we are working on in-depth info about chosen application, market application in the first phase. We are conducting more expert interviews, and we are preparing for business plan writing. And then final stage is a business plan writing, which is based on results on market analysis and contains direct clues how to enter to the market. In case of low TRL projects, we are performing single-stage market analysis with a context of markets, market opportunities, key players, situation, and then, based on this knowledge and also based on the discussions with consortium, we are writing a report about possible scenarios of exploitation of the results of the project. So, in simple words, what we will do with all results and technology, which was gained during the project. When it comes to dissemination, communication, and IT tools, it means for us graphic identity of the product, so including logo, templates of documents, promotional materials like video, brochures, roll-ups. Of course, it depends on the discussions with consortium and the needs. We are also taking care about the website of the project and newsletter. And also we can offer an internal server for very safe exchange of files between members of consortium, something like Google Docs, however, with a higher level of safety. We are also offering, together with our partner, French partner, intellectual property rights management. The main core is monitoring and guidance on IP issues. Thanks to Pacman solution, it is a special program to monitor patent opportunities. And also we are taking care about all aspects of intellectual property management in a project. So we are taking care about the IPR management. So that's what we can also do for you. So that was a knowledge about our company and how we can help you in that field. Thank you very much for your attention, and I'm open, of course, for your questions, and in case of any doubts and if you feel that you need a support in your way to go to the market, we are open for contacting. Just write to us. You can find my email address on the chat of our event. So feel free to contact us. Thank you very much.
Thank you so much, Sarah, for giving the presentation about your company and the overall support you can give to projects. And Sarah has also, they have shared their contact details. So if you want to reach out to them and include them in, you know, in the consortium, then please reach out to Sarah and her colleagues. We go to our next presenter, Sri Joydeep, from the Sri Joydeep Ashram Gyan Yoga Training and Research Center in Burdwan. We're very happy to have you with us, and we're looking forward to their presentation on immunology, developing the power of protection in the body. The floor is yours. A very good afternoon. Mr. Sri Joydeep, are you able to hear us? You have to unmute your mic. And are you able to share your presentation?
Unmute. Am I audible now?
Yes, yes, you are.
Yes. I'm trying to share my screen.
Okay.
Yeah.
Would you like us to share it, your presentation?
Yes, if you could, then that would be fine.
Yeah, okay, let me do that for you. Hold on a second. I will just share the screen. It's this one, right? This is your presentation?
Yes.
Okay, please go ahead. I will just let me know when to change the slides.
Okay. Here we are. Hello, everybody, and good afternoon, all of you. Today we are going to present something which is about yoga. Yoga is an ancient mind-body practice that is increasingly recognized to have health benefits in a variety of clinical and non-clinical conditions on pandemic preparedness and response. We have done a systematic review which have summarized the findings of randomized controlled trials examining the effects of immunoyoga, which we have introduced on immune system's functioning, which is imperative to justify the application of it in larger context. The results imply that immunoyoga may be implemented as a complementary intervention for populations at risk or already suffering from disease with an inflammatory component on pandemic preparedness and response. Now we go back to what exactly we have done on it. This is just to give an idea that what exactly the workings, how we have find out from the yoga system is that there are two circulatory systems, as you are all scientists here, we might be knowing that one is a cardiac, but I'm just rephrasing it just to make my point clear. One is a cardiovascular system and the other is a lymphatic system. Whereas the cardiovascular system has a pump like heart, but it is a closed system. In contrary, lymphatic system is open and has no pump, and that's why it only circulates 3 liters of blood, whereas the cardiovascular system has 17 liters of blood circulating in it. Lymphatic system has far more outreach and reaches the remotest corner of the body and plays an important role in transmitting lymphocytes, that is B cells and T cells, which are building blocks of immunity. Now we have found we have come right now in a sedentary lifestyle. So human body in general is is systematically made to travel, to move, that this lymphatic system functions in an optimal way. But because these days, or in 21st century, we have this sedentary lifestyle where we sit down a lot of time in front of computers and things. So because lymphatic system doesn't have a pump to pump it like the cardiovascular system had, and lymphatic system, the outreach happens when we really move, So what we have tried to with our immunoyoga, what we have tried to move is that we have tried to create certain steps which helps you to move the body. Like there are, Viparit Karana Asana, there is different mudras, there is Shirshasana. These helps you to move the lymphatic system in a different way, right? To make the circulation optimized, that the B cells and T cells reaches in the remotest corner of your body. So these particular thing we have developed, we have implemented to the students in the pandemic time, before the pandemic, and we have seen that this creates great results. Their immunity develops because they are now able to move the entire system. These are standard poses. So this immunoyoga acts like a pump, and it actually helps the body, and it actually helps the lymphatic system to be energized. The sequences and practices creates better outreaching of transmitting lymphocytes, B cells and T cells. Now, this is what we have done, and a certain form of relaxation arrives with the lymphatic mode activation through immunoyoga that needs to be examined. Now, this is what we have done. We have seen that this works, but probably this thing, what we have done, if you look at it, it's a complementary therapy. One of the benefit, you are not putting any fluid inside the body, you are just positioning the body. It has zero side effects. So even if you have the best of the vaccines, best of antiviral therapy, you will have some kind of side effects. But here you are just moving the body in certain optimal positions, and that is activating the lymphatic system. So you have zero side effects. And at the same time, there are certain experiments we have come to know that has been done on different parts on, Mysore and other, where there are instruments built where people who are unable to get them into that position, such instruments like, Viparit Karini Mudra, which is Viparit Karini Mudra is one of the very good effect where the lymph nodes, basically we have lymph nodes in tonsils, we have lymph nodes in spleen. So these lymph nodes becomes activated, so that Viparit Karini Mudra, a persons of senior age, it would be difficult to do. So there are, instruments built like a walker, where you can go there, and in the instrument, when you put yourself, it's like you put for 1 hour or certain point of time, and you get that lymphatic stimulation which will require for the B cells and T cells to travel on the remote corner of your body where maybe the viruses are existing, and where this immune system can get, and its body's inner potential to fight back to this entire whatever the pathogens are doing to fight back. So this is what we have seen, and we wanted to do further research on this area. We have seen that 21 days is a very good time to create a habit. So whatever the intervention, the yoga, the complementary therapy intervention, we suggested to do it for 21 days, and we see that it now becomes a habit on the person, on the subject, and so it can continue over the lifetime. And we are at Accent Heritage Institute. We are an NGO partner of United Nations Global Compact. So we wanted to scale and launch this program. We wanted partners, and we wanted to go to the different children and adolescent community who have lesser, the strength of their immunity is less. So we wanted to reach out them. So this is why we wanted to have partners, and this is what we are trying to do here. we are looking for further research in Immunoyoga program in its role of lymphatic system activation for preparing proposal in the call Horizon Health 2023 Disease 034, and we are looking also for partners on dissemination outreach of Immunoyoga program and making better outreach to vulnerable communities across the world. So this is what our entire objective towards this presentation is. My email address is joydeep.chakladar@gmail.com. If you think that we can collaborate and we can work together and we can give a good value, because this might be indigenous knowledge, but this indigenous knowledge has a big scientific application and it could be applied across the world. And so if you think that we can fit in, please do write to me, joydeep.chakladar@gmail.com. Thank you very much for giving me this opportunity to present here.
Thank you so much, Mr. Sri Joydeep, for your presentation and reaching out to potential partners for this very interesting proposal, project idea, using yoga and scientific methods for strengthening the immunity of the human body. Shri Joydeep, you also should put your contact details in the chat because it's not available on the slide. So kindly do that in the chat. And we go over to our next presenter, which is Dr. Jitendra Singh from CSIR, Central Electronics Engineering Research Institute, Pilani in India, with the project idea development of biosensor electronics platform. Very good afternoon, Dr. Jitendra. Floor is yours.
Hi, sir. Good afternoon. I'm audible?
Yes, you're audible and visible.
Yeah.
Your presentation.
Thank you, sir. Thank you, sir, for this opportunity, and thanks to the organizer for this event. We are a part of a CSIR and a CSIR laboratory, Central Electronics Engineering Research Institute, located at Pilani. And we are an electronics engineer. So we are looking forward that how this electronics platform can be useful for the biosensing applications. So this presentation will be about that. So devices we are working, development of biosensor electronics platform. So our device will be surface acoustic wave resonators. So resonator, this figure is visible here. It is a electronics resonator, which is based on the piezoelectric materials. So it has one transducers, conductive IDT transducers, which is used to convert the electrical energy to the acoustic energy and back. Standing waves are produced and established on the surface. So it has two transducers, and there can be one reflector and the other reflector to absorb the acoustic waves. So it is the basic working of these electronics resonators. Antibodies are in proper orientation and that can react with the target biomarkers, so they can be oriented. Analyte binding events can happen on the surface, and this can be translated into an electronic signal. So this biosensor can be useful for various applications like biomarkers in the blood or in urine or in serum or saliva. So this device can have the various applications, and the biosignals we can convert into an electronic signal which is directly measurable. So if you see this sensor, it is a frequency-based sensor, and we see a frequency change when specific event happen on a sensor surface. So either the frequency will shift or phase will shift or signal will attenuate. So that electronically we can monitor, and the resonance frequency is decided by the acoustic wave velocity and the wavelength of these resonators. So any event happens on this sensor surface that will change the acoustic wave velocity. So any analyte binding event can happen on the sensor surface. Any chemical specific event or reaction can happen on the sensor surface. Any mass change can happen, or stiffness will change, or its conductivity will change, or due to some temperature or some viscosity will change, or some pressure will vary. So this sensor platform can convert the events into an electronic signal which is directly measurable. So it can be a very useful platform to make out these biosensing devices, and this can have a unique feature like the point of care applications. It is very fast. Within a few seconds, this gives the response, and highly sensitive. Picogram level of mass change can be possible with this platform. Very specific. It is label free, disposable, and the mass production is possible. It is easy to use, and it is not so expensive when it is batch produced, and easily accessible, highly selective. It can be made by the proper functionalization, so it can be used for in the screening devices or point of care devices. So we are looking for the partner which has the strong background in the relevant field, have the biological sensing or the chemical sensing for the specific target applications, and skills in the functionalization of the surface assembled monolayers for the biolayers or functional layer for the specific applications, immobilization of the antibody or antigen on a specific orientation for the specific target molecules, and having a strong understanding of the bio or chemical events happening on the sensor surface, and very keen to work for the this electronic biosensor platform. Thank you, sir.
Thank you so much, Dr. Jitendra, for your presentation. And we move to our next presenter, which is Dr. Amit Awasthi from the Translational Health Science and Technology Institute, India. Dr. Amit, very good afternoon. I hope you're with us. Yes, I can see you. And Dr. Jitendra, could you kindly stop sharing your screen? Thank you.
I'm doing so.
Thank you.
Thank you very much. I'm not able to share my, you cannot start.
Yeah, I think Dr. Yeah, hold on one second. Yeah, now it should be possible, Dr. Amit.
Okay, great. Thank you very much. So you can see the screen.
Yes.
So thank you very much for giving me this opportunity to present. So some of the things which THSTI and my lab and Dr. Sweetie Sumal, who is not on the call, has developed models which can be, partnered with upcoming Indo-European grants. So this is something which is COVID-19 research, what THSTI and my lab has contributed, funded by DBT BIREC fund under the mission COVID Suraksha. So what you can see, we have done multiple things in the COVID-19. First is the characterized COVID, SARS-CoV-2 and VOCs, as a reagent. So we have this viruses as a repository. We have clinical isolates. We isolated some of these strains in India and cultured and made repository. Using the viruses, what we isolated, we also developed animal models, both hamster and K18H2 transgenic mouse model, where to study the vaccine efficacy and various antiviral screenings using animal models. Virus pathogenesis. So using this animal model, at the same time, we also try to understand virus pathogenesis and immunology around the viruses, how this virus causes the lethal, condition in hamster as well as human H2 mice model. And obviously we have biorepository, developed biorepository samples from, COVID convalescent individuals or post-vaccination. We have PBMC and various other sera samples to do some of the human work, particularly standardized T-cell assays, which become very handy for, understand the, various VOCs, circulating, and how this T-cell response can actually contain the virus in the condition when antibody response becomes suboptimal for VOCs. And obviously next generation pan beta coronavirus vaccine development. And for this, we have received around 12.5 million. This is wrongly written, million USD with THST and Panacea from the SAPI. And the idea of developing this pan beta coronavirus vaccine, that, we have a platform in which we are actually stitching different epitopes of SARS-CoV-1, SARS-CoV-2 and VOCs, as well as MERS. So this platform is now at the stage of preclinical validation, and hopefully within next few months, we will have the preclinical validation of the platform, what we have received the funding from the SAPI. So using this various, platforms, what we have generated, using our platform, we have conducted preclinical, clinical trials for COVID-19 vaccine, antiviral drugs, and therapeutics for various biopharma industry and academia in India. In addition, we have carried out basic immunopathology research describing the virus pathogenesis and host responses. So in addition to this, so using this platform, what I have shown in my previous slide, we were kind of supported very indigenous vaccine like biological E. Corbevax. I am very happy to tell you that all the clinical samples for antibody neutralization as well as T-cell response for this clinical trial is done by THSTI and my lab, where this data we have presented to DCGI, and subsequently this vaccine actually given emergency use authorization. We tested the first DNA vaccine of the world for COVID-19 in hamster model, and we have provided the preclinical data to government for its further clinical trials. We also tested the worm COVID-19 vaccine developed by Minvax, Bangalore. In addition, the Bharat Biotech, we have done the work for Covaxin as well as some other vaccines we have tested in preclinical and clinical models what we have generated. These are the few publications what we have done for COVID-19, and in 2021 we have published paper in Lancet Infectious Disease where we've shown how T-cell response remained preserved during Delta surge in India, and vaccine effectiveness of Covishield remains 63%. Some of the work we have done in golden Syrian hamster, where we have actually identified how SARS-CoV-2 infection lead to the cardiovascular dysfunction and this virus actually goes into cardiomyocyte and actually create a problem in the heart. And there are other papers which is already you can see, you can go online, and if you are interested, I will be happy to share the print or preprint of these publications. So work which is Dr. Sweetie Samal and part of my lab is also working on this. We have 3 major grants from the Indo-European Consortia. You can see the incentive, ENFLU and Indigo. These grants, the whole idea to have these grants to develop a next generation flu vaccine. So this is something well-characterized virus repository we have. We have next generation vaccine development platform. We are testing preclinical animal model, both mice as well as ferret model. We developing or developed virus pathogenesis as well as immune response for flu viruses. So in the virology program is an integrated platform to fast and efficient screening and pathogen progression of vaccine antiviral for virucidal coating. Our capability range from screening to animal model, availability of novel tools assays. So basically creating virus repository of influenza A and V viruses. Research activities focuses on discovery of biomarker, resistant strains and viral references, preclinical challenge strain development. Viral in vitro assays, plaque we have for influenza, we have plaque assay, hemagglutinin assay, ELISA, protein antigen, inactivated whole virion, as well as we have neutralization assay at already developed in THSTI. Animal model, as I mentioned, that we have both BALPC and the B6 mice model, which is a gold standard model for evaluation of vaccines and inhibitors. We have ferret model, which is a gold standard model mimicking human infection. This is under development. So using these models, we are in a unique situation to support the next call for Indo-European Union to have a preparedness for antivirals. In addition, this is a ferret facility which is inaugurated almost a year ago. Now the support human respiratory virus research in India, first in India to support evaluation of next generation influenza vaccine candidate, open to all researchers, academic and industry. So this is, we are making a national facility. Anybody who would like to use this facility, we will be very happy to support. In addition to influenza and COVID-19, we also have preclinical model for other viruses, particularly arboviruses. This is the AG129 model where, all type 1 responses are actually, type 1 interferon responses are not there. So this mice become very susceptible. So AG129 deficient mice in interferon alpha, beta, and gamma receptor type 2 interferon, completely competent in eliciting humoral immune response, exhibiting dengue virus or JV-induced pathogenesis when infected either intravenously or intraperitoneally with challenge virus. The important clinical characteristic feature of DNV virus infection, the vascular permeability is observed in this model. As you can see on the slide, the typical JEV, Japanese encephalovirus virus induced neurological symptoms are seen in AG129 mice infected with intraperitoneal injection of JEV clinical isolate. The model has been validated using an adenosine nucleoside inhibitor of DNV. The model is suitable for evaluation of novel vaccine candidate, antiviral drug, and therapeutics. So with this, now I actually presented you our overall capacity, which can support any partnership with any European, or multi-consortia grant, where we can provide our expertise, not only for the vaccine, but also for antiviral testing in preclinical and clinical model, because we have most of the assays has established and developed, and we have shown in different disease settings. So if anybody interested, we are happy to collaborate. And any questions regarding to this, I will be happy to answer. Thank you so much.
Thank you so much, Dr. Amit, for presenting what kind of research you've been doing, and also the EU-India partnership, where you have been very, strong in the last calls. Now we move to our next presenter, Dr. Santosh Bothe from the Principal Saraswati College. A very good afternoon to you, Dr. Santosh, and the floor is yours.
Thank you for doing this.
Dr. Santosh, you have to, there's some issue with your mic. If you could maybe either get closer to it or try to.
Can you hear me now?
Yeah.
So I'll try to speak a little louder. Okay, I'll be presenting my idea of monitoring the various measurable biosignals non-invasively and using it for the pandemic response as well as surveillance of the various diseases. So the potential biosignal which I'll be targeting is idea of the voice, and it is being commercially used in one of the Italian company called Voice, wherein we can measure the diseases associated with according to the statistical sound. It has proved, I will be sharing few reasons. Then smart ECG, even the optical images of the face. We have developed around 136 templates of the various skin textures, like Asians or different skin texture, European, American. So we have 136 skin templates. Remains, we call it as a normalized image, and that normalized image is analyzed. System, and the diseases associated with the system. So things which are affecting the respiratory systems. The pulse is big. This piezoelectric sensors, what we are doing is we are analyzing the 1,000+ characteristics of the pulse, and those characteristics are used as the digital disease markers. And finally, the motor moment. Here are the few results which I'd like to share with you, particularly for voice. Dr. Samrat, hope I am audible.
It's a bit off and on, but if you hold it closer to you, then it's better.
Okay, is it better now?
Yes, yes.
Okay, so in the voice recording system, there is a predefined script which person has to read, and then we have a pre-processed filter which normalizes the person's specific characteristic. These are 12 characteristics, and we record total 6,300 plus voice characteristics. These voice characteristics are subjected to the pre-trained AI-based algorithms, which establishes the relations between the characteristic changes in the voice parameters and the underlying health conditions. So here are some cases which we identified. This is what we call a digital marker for the diseases. So here I'm showing the 4 parameters like P1, P2, P3, P4, and then the healthy range, and when the person has a pulmonary tuberculosis, how it changes. So we have tested on 360 patients, and it's good sensitivity and specificity as well. Besides this, the optical face image can be used to measure various characteristics, as I explained earlier. So here are some samples which we have measured using the optical face image. Particularly the endocrine system reflects on the face if you talk with the clinic. And we have some results to do that. It is all the 3 techniques which I presented here are protected with IP in India and some of the European countries. So overview, we are like to use this biomarker. So why I call it as objective analytics, because subjective analytics, we have predefined process and there is multiple protocols to be placed. But the objectively looking at the whatever we can measure and understanding what are the patterns which are for the specific health conditions can help us to monitor a pandemic or epidemic in real time. And these sensors are sensors which can be used to measure this, are quite handy and connected with the smartphone. And this will help us to monitor the overall population for the particular epidemic or pandemic condition. The possible collaborators, which we have some earlier had some earlier discussion or some earlier submission to the Horizon or other funding agencies. Principally, they are not aggregated, but we are talking to them. They are our potential partners. The Pravina Adoni, who is working on developing various sensors for biomedical applications. Vita Viera from Innoadi, Germany, they are having expertise, complementing expertise. So we'll be talking to them, and we are looking for the clinical partners. If at all everybody agrees, we'll like to form the consortium with these many members. Thank you very much. That's all from my side. Thank you.
Thank you so much, Dr. Santosh, for your presentation. And we have one more presenter today, Ms. Vidya Desai. I hope she's with us. Yes. Kindly ask you to share your presentation on conceptualizing the need for antiviral therapeutics for sustainable future. Thank you. And kindly unmute your mic as well.
Thank you. Can you see the presentation?
Yes, ma'am. Just if you could put it into full screen mode, that would be ideal. Perfect.
Very good evening. Myself, Professor Vidya Desai. I would like to thank Horizon Health, Horizon Europa for giving this opportunity to me, and also the Indian who are heading this particular call on this pandemic preparedness and response, broad spectrum antiviral therapeutics for infectious diseases with epidemic potential. I would like to give a brief profile about the proposal which I would like to make for this particular call, that is designing protein-linked heterocyclic scaffold as small molecule virus inhibitors for as with synergy potential. I come from a very small state, that is Goa in India, and and I come from a very small research center. And when I saw this call about looking for a consortium for your work, research work, I was very happy. It has thrilled me a lot because it was way back 2 years back when the COVID pandemic had hit. That time I was looking for a partner for my molecules to be tested for antiviral activity, but I was not able to find one. It so happened that I could get a partner at IIT Guwahati, so that I would like to also show in why I entered into this antiviral field. So this is our small institute, and this is my trust area which I work on. Basically, I'm an organic chemist. All the presenters have thrown light on how they could use detection tools for viral disease. Here I would like to talk about the actual molecule which I would design in order to make a viral therapeutics, which could be a broad spectrum one. So I mostly work on green and sustainable synthesis of heterocyclic scaffolds. As we all know that heterocycles play a very important role in designing drug molecules. My work area is basically on infectious diseases. First, I have been working on tuberculosis, and wherein we have designed inhibitors for vinyl ACP reductase enzyme. Then we are also working on tyrosine kinase inhibitors for treatment of cancer. The work on benzodiazepines has been filed for a patent already, and designing COX-2 inhibitors for treatment of inflammation. And then I entered into the field of antiviral activity to find antiviral therapeutics. Herein, what happened is that I had designed some molecules, pyrozoles, okay, and these pyrosol molecules I wanted to do repurposing for COVID-19, but I could not get the right partner to make these molecules effective for the virus. So what I did is that I tried to email many people, and then finally I got a collaborator in IIT Guwahati, and I designed these molecules were tested by that professor from IIT Guwahati, and compounds was showing very good potency against JEV, that is Japanese encephalitis virus. So in vitro and in vivo studies were all done for this compound, and so this particular work appeared in virus research. So that has given me a lot of the encouragement to work towards designing antiviral therapeutics, and we have been designing now labs many different type of molecules having different moieties, heterocyclic moieties. So basically, this particular proposal, I would like to focus on flavivirus. And since we have already done the studies on Japanese encephalitis virus, we want to focus on such other viruses like dengue virus, then Zika virus. And another potential for this is that because these particular viruses have no drugs available, so therefore we need to design such antiviral medicines for this. And we have seen certain heterocyclic moieties like pyrozole and hydroxyzole and all. These particular molecules can be very good potential lead compounds to get developed into an antiviral agent. And also, like, we could also get some novel mechanisms out of it, how to kill these viruses. Another important fact is that most of the presently known antiviral drugs which are there, they have been used as a repurposing for this, drugs, these particular viruses to fight against these viruses. But then we could also try something wherein this particular molecule, which will be designed for this proposal, could be used in synergy with such antiviral drugs, existing drugs, and that would give a better address the issues related to antiviral need. So this is the work proposal wherein we are targeting flavivirus, and these are the heterocycles we would like to work upon, because I've worked on more than 25 heterocycles in my short career, and this is what we would like to focus on, having a proline based heterocyclic conjugates. Okay, proline being a very important part of, in case of dengue virus, it's the protein which is having which is very rich in proline. So this particular concept would help in designing such molecules that would help us in making a broad spectrum antiviral therapeutics. So further on, since we had also worked on pyrozole compounds are the 5-membered compounds. those compounds which we have worked on, they were showing like fluorine-based compounds were showing better activity, and it was showing very promising results were obtained for that particular those compounds. So we are having a different particular strategy for this, wherein we could design some different functionalities having fluorine substituents on pyrazoles and different other moieties attached to it, and that could be a very good proposition to treat diseases caused by flaviviruses. So this is what is thought about. So all what is needed for these compounds are, because we are only synthesizing the compounds and synthesizing in a novel way. So that work is done in our synthetic lab. But what we are lacking is getting the in vitro studies done, assay studies done, and the in vivo assays done, and also of course the synergistic study. So for this studies, we need to have some partners. So one of the partner we have is an Indian partner already, that is Professor Sachin Kumar from IIT Guwahati, with whom we have successfully worked on this particular project, and we have got a paper published in Virus Research. So what is the novel thing about this particular proposal, which will be put forth for this particular call? We will be designing novel compounds, because such compounds, we have a chalcone moiety attached with a proline-based heterocycle. Different heterocycles could be linked to, so that we could have a very important structure activity relationship studies. And also the synthesis would not be a normal synthesis, it would be sustainable synthesis. We could use renewable resources, we could use other sustainable technologies for designing these compounds, and that plays a very important role in the economy point of view. So then this particular strategy which we would like to propose is for the first time, wherein there is a linkage between proline, chalcone, and heterocycles. So, and this further addresses the crisis which has been growing against for the rise of flavivirus diseases. And so this can help us in countering such viruses. And further on, synergistic activity with the existing drugs is also a novel approach in order to get a better broad spectrum antiviral therapeutic. Now, what we have right now with us is that we have a synthetic organic chemist and green chemist to work on the synthesis of the target compounds. We also have a computational chemist to get us the molecular docking scores, and also biologists to work on maybe an in vitro in India antiviral assay. But we would want another partner from internationally acclaimed we need to identify novel mechanisms through various antiviral, because the compounds which will be synthesized will be very novel based, having different type of moieties, heterocyclic moieties, as well as it would have a chirality component in it. And also in vivo antiviral model is the search we would like to have in a partner for in an international arena. So this is already we have in our labs. So this is the expected outcome. A novel proline chalcone heterocycle conjugates will be synthesized, promising antiviral assay results with molecular diversity, and synergistic effect of these compounds for effective antiviral medicines, promising strategy towards broad spectrum antiviral therapeutics. Thank you.
Thank you so much, Dr. Vidya, for your presentation. And we are now actually at the end of our flash presentation session, but we have still a few more minutes, so we could use them now for some specific questions to the presenters, or maybe the presenters among themselves would like to ask something to the others who have presented. So we could take a couple of minutes on that if you like. Also ask Vivek, Dr. Vivek, to also join into that conversation.
I think there is no questions, Samrat. We can go for a conclusion remark by Dr. Abhijit Singh from DBT. Abhijit, you are online.
Okay, yeah, then I see Dr. Abhishek is there. Dr. Abhishek, could you kindly open your video and
Hello.
yes.
So actually, I have logged in from two other systems because there is some issue regarding internet connectivity. That's why. So hopefully, my voice will not break and got interrupted by the network fluctuation. So first of all, I will thank to for organizing this brokerage event, specifically for this call. So in my view, the pitching event for this call was very informative, since the 7 presentation along with the brief information about the call was provided through various presentations. And in what we think, along with the WHO priority list for the antiviral therapeutics, as well as for the detection of the virus, as well as to find out the ways for therapeutics, along with the new innovative technologies that can provide in short term as well as for the long term detection methods, treatments, as well as the repositories for the welfare of humanity. Since like as we have seen, specifically in India, like Kerala route as well as for the Chennai route are very vulnerable for the viral diseases, which generally got reported from the African area, whoever is getting traveled from African continent. So these things should also be incorporated from the Indian applications. So that will provide the broader spectrum for the coverage of pandemic preparedness, because we don't know when the virus comes and when which infection route or transmission route that can provide the vulnerability to the human. So this call is specifically, which I have gone through, that they specifically for the human. So I hope we will get very high traction for the submission of applications with the interdisciplinary and intercontinental application, likewise covering more than 2 or 3 European Union countries, as well as public and private participation of organizations along with research institutions. And my special thanks to Vivek ji, as well as Philon, and for Samrat, as well as for all participants from Indian side, as well as from European Union side. Thank you very much.
Thank you so much, Dr. Abhishek, for the very nice concluding remarks. And we are really looking forward now to, and crossing fingers that good consortium will be formed and we will see some very interesting applications. I would like to invite all the presenters and speakers to come back online with their cameras for a final screenshot, for our records. That would be wonderful to have you all with us. Yeah, I see Dr. Abhishek and everyone is coming on. Okay, give it a big smile. Fantastic, thank you so much. Thank you to all of our participants who have been joining and listening attentively to this more than 2 hours event. And with that, we will conclude for today. We will upload the contact details of the presenters on our website, and of course also make the recording available to you in a day or so on our YouTube channel. And wishing you all the best and have a nice afternoon here in India and good lunchtime in Europe. Thank you so much for joining. Bye.
Thank you. Bye.
you. Bye bye.
Thank you.